We are beginning the session 1, Biological Methods for Waste Treatment 1, session chair Professor Dr. Ince.
Thank you very much. Rosa.
Hi, good morning.
Hello, good morning. Good afternoon.
I'm sorry, I was trying to solve some technical problems.
That's fine. Please go ahead your presentation. With your presentation, please go ahead. If you
okay. Just a minute. I will share the presentation. Just a minute. I recorded the presentation. I'll try to share just a minute.
If you finish your presentation in 10
minutes. I'm opening the. Can you see my screen?
Yeah, I can see the screen.
Okay. Okay.
Okay, we can see your presentation. Are you ready? Mr. Rosa? We can start.
What happened?
Maybe you can full screen. You can do full screen maybe.
What?
You can use full screen option maybe.
Can I restart?
Oh, that's fine, I think. Yeah, that's okay. You can
it's
go
okay.
ahead. Yeah, you can go ahead.
Okay. Sorry.
That's fine. That's really good.
Okay.
Is it a video or presentation? I think, okay, that's presentation, right? Mr. Rowe, I think there is no sound.
No sound?
Yes, there is no sound. You need to...
because yesterday I did a test with the Congress and everything was okay.
Yeah, you need to switch on your speaker, your computer speaker. You need to switch on your speaker. Or maybe you can present yourself.
Just a minute. Is it possible to heard?
If it is possible, you can speak about the presentation if you want, or if it's okay.
Okay. It's impossible to heard.
If it is okay, you can share the information with yourself.
Okay,
Okay.
I'll share the presentation. Just a minute. Um, Apresentar ao vivo do problema. Me ajuda. Can you hear me?
Yeah, share the
Okay.
presentation.
Okay. Thank you.
Yeah.
Good
You
morning,
can start.
everyone. Good morning, everyone. I'm sorry for the problems, technical problems. My name is André Rosa. I'm professor at Federal University of Viçosa in Brazil, and I will present the article entitled "Which Mathematical Model Best Fits the Methane Production in Pig Farms with Covered Lagoon Digesters." The other authors are Josiara Oliveira Lopes, Alisson Carraro Borges, Isabela de Paula Sousa, Silas Modesto de Melo, and T.Massuger. We have in Brazil the agribusiness, that is one of the most important sectors of the Brazilian economy. And in this the sector, we have the pig farming that produces a high volumes of manure, which contains a high content of organic matter, nutrients, and metals. And in Brazil, we have widely used of covered lagoon digesters that we can see in this picture. And using the systems, we have some benefits as such as the lower implementation and maintenance costs, as well as the biogas energy recovery. The biogas production in the system, we can analyze or try to estimate this biogas using the mathematical models. But we have an absence of validation for full-scale plants in specific conditions, such as climate, digester design, among others. And in Brazil, we have some methodologies and international methodologies that we can use to compare or try to estimate the biogas production, as Shane, mass balance, CETESB, that is a national methodology, and a methodology from Intergovernmental Panel on Climate Changes. The models require adjustment input data, being scarce studies for model validation, especially in our situation that are full-scale plants in warm regions, such as in Brazil. And that there is an importance of mathematical model for the biogas production that can help in this decision-making process and the improvement of sustainable energy in pig farms. This study aimed at evaluating which mathematical model best fits the methane production in pig farms operated by covered lagoon digesters. Here we have a full shot of the system. The system is in Teixeiras, a town in Minas Gerais, Brazil. The biogas is recovered from covered lagoon digester and converted into electricity using internal combustion engine. And this electricity is inserted into the grid. And the monitoring period was from September 2018 to August 2019. And we have some sensors installed in the system that are transferred to a laboratory where we can manage the data. The input parameters from these methodologies that we compared were number of animals, percentage of methane, biomass temperature that was collected in the sensors, chemical oxygen demand, and volatile solids. Here you have the equations from the systems, the mathematical model of Shane with the input data in red, mass balance, the details of the methodology, CETESB, and IPCC, where the main input data is the total number of animals. In these methodologies, we don't consider the characteristics of the materials, and when this happens is with theoretical values. The measurement of biogas composition were performed using a gas analyzer. A Manure campaigns were carried out on a weekly basis, following the standard methods, and the Manure flow, the water consumption, and the Manure flow was estimated by the using of water consumption and a coefficient of 65% of conversion from water to Manure. The actual methane production, that is the reference, the value that we use to compare the methodologies, was monitored during this period. And here is the how we calculate the actual methane production. In this case, we used the electricity production and at the end, the methane production in the system. And the P, the production actual, and the mathematical models were compared by using t-test for a significant level of 5%. Here are the main results of the research. The monthly average of animals and effluent flow. Here, the number of animals, the variation, and the effluent flow within an average of animals. Okay?
Yeah, okay. Okay, go ahead.
Okay. Around 11,000 animals with an effluent flow 102.3 cubic meters per day. Some results of monthly average of COD, volatile solids, with the variation through this period, with the concentration. And the methane production, monthly average of percentage of methane in biogas and internal temperature. Here the percentage of methane, that ranged from 43.6 to 81.9%. And the biomass temperature inside the COD varied from 20.1 degrees to 26.8 degrees. An exploratory evaluation of the system, comparing the methodologies. We have the methane production, the box plot of methane production for evaluated models, and the monitored system for Shane, mass balance, CETESB, IPCC, and the reference of the system, P actual. According to these results, we can highlight that Shane and IPCC models showed similar results in comparison with actual data, in this case, P actual. About mass balance, we obtained an unsatisfactory performance of mass balance model, and this could be associated with the flow rate variation, the COD variation, and methane losses dissolved in the effluent that were not taken into account in the original model. About CETESB, the model presented a discrepant result. It considers an input data, the number of animals, and some theoretical data. In this case, the unique input data is the number of animals, and the other values are theoretical, and does not consider the main variables reported in the literature for biogas production. The statistical study using t-test to analyze methane production for the models we can observe that the Shane and IPCC models did not present any statistically significant difference in comparison with actual data, in this case, monitored system. So these are the most important and representative methodologies to represent the methane production. On the other hand, the CETESB and mass balance models indicated statistically significant difference with significant level of 5%. So we need, for example, try to adjust or to propose some alternative methodology to improve these results. The main conclusions of the work: the Shane model presented a satisfactory performance and allows evaluating the methane production in the time series. The IPCC model also presented a similar performance, but it was not sensitive to peak frame variations. The mass balance underestimates the methane production, while the CETESB model overestimates the actual data. Both models indicate differences between the actual data, and the improvement of mathematical model based on full-scale plants need to be studied in order to produce more reliable information that could be used in projects to recover biogas. The authors would like to thank the financial agencies in Brazil, CAPES, FAPEMIG, and SENFEQI. And here are some references of the presentation. Thank you for your attention.
Thank you for your presentation. I would like to ask your questions. Do you have any question? You can write in the chat section if you want, or maybe you can ask with speaking. If you don't have any question, I will skip the other presenter. Okay.
Okay, thank you.
Thank you very much. Our second presenter is Emine Gözde Ozbayram. Emine Gözde Ozbayram graduated as an environmental engineer from the Environmental Engineering Department of Istanbul University in Turkey in 2009. She received her Master of Science degree in 2012 and PhD in 2018 in Environmental Engineering from Istanbul Technical University in Turkey. She works as an assistant professor at Istanbul University Faculty of Aquatic Sciences. Her presentation title is Co-inoculation of rumen fluid with methanogenic sludge exhibits distinct microbial communities in semi-continuous single-phase and two-phase anaerobic digestion of cow manure. Ms. Ozbayram, microphone is yours.
Yes, thank you so much for the nice introduction. I'm sorry about the long title. I would like to thank to the symposium committee for selecting this study and giving me the chance to share the information with you. I will start with the brief introduction. As you know, biogas production from renewable energy sources is its increasing trend in Europe actually, and over 17,000 biogas plants across Europe is producing electricity for almost 50 million European households. And besides constructing new plants, they are dealing with the focusing on the more resilient and productive operation. And when we look at the feedstocks, we can say that cattle manure, agro residues, organic waste streams are more sustainable substrates for anaerobic digestion, as we know. And in the developing countries, we heavily rely on the fossil fuels from abroad. On the other hand, we have so much benefits, so much abundant lignocellulosic feedstock that we can benefit as the biogas production. And we would like to have that kind of lignocellulosic feedstock for anaerobic digestion because their abundance, their low price, and relatively high methane yields compared to the other feedstocks. But as you know, during the anaerobic digestion process of the lignocellulose-rich feedstocks, we have a problem with the hydrolysis step because of their complex structures. Let's have a look on the manure. Almost 50% of the animal manure is composed by the lignocellulosic fibers like cellulose, hemicellulose, and lignin that we should deal with their complex structure. And the anaerobic digestion of the animal manure is a sustainable solution. On one hand, the renewable energy production, on the other hand, waste management. And when we are talking about the anaerobic digestion, the balance of the 4 steps of the anaerobic process needs to be in balance, and any kind of inhibition on any steps can affect the biogas efficiency at the end of the process. And when we look at the nature is always an inspiration for engineers or scientists, and we can say that ruminants can deal with the lignocellulosic feedstocks very well. They have unique, sophisticated digestive systems that they can metabolize lignocellulosic components easily. And when you look at the literature, the rumen fluid directly used as the inocula in the anaerobic plants, but sustaining enough inoculum from the rumen fluid is not that possible actually. Then we can consider the selective enrichment. They have cellulolytic bacteria inside their stomachs in the rumen fluid, and we thought that they shared similarities with the anaerobic reactor and the rumen fluid, like temperature, pH, and other components. And we thought that selective enrichment can be a strategy to produce more effective inocula for the anaerobic digestive systems. And our motivation in the study was coming from our previous studies. In the batch experiments, we saw that the bioaugmentation with the enriched culture from rumen fluid can be an effective solution for the anaerobic digesters treating lignocellulose-rich biomass. And here, the aim of this study that I'm presenting today is to compare the co-inoculation strategies in the semi-continuous digesters in 2-phase and single-phase systems, and compare the results, and of course, and also the show the dynamics of the representatives of cellulolytic bacteria inside the systems periodically. For this purpose, we collaborated with Istanbul University Veterinary Faculty, and we took our manure samples and rumen fluids from Istanbul University, from a non-medicated healthy Jersey cows. And I'm not going to give you the details about the enrichment procedure, but we done the enrichment under anoxic conditions by Bushnell house medium, using Bushnell house medium enriched by the carboxymethylcellulose. And after a couple of transfers, we got the enrichment culture, and we inoculated the digesters from standard inoculum that we obtained from full-scale treatment, full-scale anaerobic digesters treating manure from Bursa. And these are the simple characteristics of the sea sludge and our rumen fluid and cow manure. And I can say that they have the similar features with the data compared to the literature, and we didn't face with the extreme characteristics. And when I tell you the digester operations, we had 2 different systems running duplicates actually. And in the first system, we have single phase reactors. The SRT was 25 days, and we operated the whole digesters for 3 SRT, like which means 75 days. And we inoculated the single phase reactor rumen with rumen fluid, enrichment culture, I call it like bioaugmentation culture, and also standard inoculum that we took from a full-scale plant. And in this two-phase reactor, we had acidogenic reactor and also a methanogenic reactor. The acidogenic reactor was inoculated with the Rumen fluid and enrichment culture, but we add standard inoculum that we took from full-scale plant to the methanogenic reactor to have methanogens in the system. And in every 5 days, we fed the reactors. That's why we call it like semi-continuous reactor. And the organic loading rate was around 3 gram VS per day, per liter per day. And we checked the microbial community structures in the Rumen fluid, enrichment culture, manure, and also in the standard inoculum. That's why we used next generation sequencing techniques. And after that, we analyzed the data by ourselves in-house scripts with the bioinformatic analysis, make it the data meaningful. And we also use our studio to compare the microbial community profiles in the in our samples. And we tracked the cellulolytic bacteria with qPCR. We isolated DNA and then we performed qPCR for 3 representatives of the cellulolytic bacteria, which are commonly mentioned in the literature. And of course, we use our studio program for preparing the RDA graphs to make the data more meaningful. And here, as a result, you can see the Rumen fluid that we took from the cow, and then we performed enrichment studies. And I can say that the community profile changed significantly after the enrichment procedure, and we managed to increase the abundance of Ruminococcaceae, which is an important, includes the important species for hydrolytic bacteria, lignocellulitic bacteria actually. And in the anaerobic digester, the community's representatives are belong to the Firmicutes, Bacteroidetes, and VVE1, which is quite similar with the literature data. And here we compare the communities with the NMDS plot. As you can see it in here, Rumen fluid microbial community is quite similar with the enrichment culture, but a bit different. But it is significantly different from the manures enrichment manures microbial community, and also it differs from the inoculum. So it has a unique structure in terms of microbial community. And when I continue with the biogas production results, I have to mention that we just make the inoculation at the beginning of the study, and we didn't repeat the inoculation during the operation period. That's why we had better biogas production at the first 20 days of the operation period, let's say 15 days of operation period. And then after a while, as you can see it in here, the biogas production stabilized. And at the end of the study, we had higher production in the two-phase system, but it's compared to the beginning of the study, it's a bit relatively low. And here you can see the methane yield that we calculated for the entire study. As you can see it in here, the two-phase system, we had higher methane component in the biogas, but and I can say that we had higher methane yields in the two-phase system, which is in the line with the previous studies with the literature, regardless of the inoculation strategy. I have to mention that. And in the two-phase system, the volatile fatty acid production reached around 1,500 milligram per liters, but there was no accumulation. It is produced immediately after we fed to the methanogenic reactors. And here you can see the quantification results of the qPCR. And I can say that there is a variation in the digesters. In the two-phase acidogenic reactor and single-phase reactor, we had more lignocellulitic bacteria, but also we detect them in the methanogenic reactor as well. And most probably they come from the feed, or they have the local microbial community inside the reactor. It's mostly possible. And here the bubbles show the same data with the bar chart for each period that we took sample and quantify the results, and the vectors showing the reactors. As you can see it in here, I told you that we operated the reactors in replicates. Replicates show similarity in the reactors, and we can say from this graph that the hydrolytic bacteria are more effective for sure for the acetogenic digesters and single phase digesters, but they are not that similar with the methanogenic digesters. We have them in the digesters, but the profile is quite different, and the vectors are going to the different directions. And as a conclusion, our results show that the co-inoculation strategies with the enrichment culture, with the rumen fluid, had the higher biogas production in the first SRT in two-phase anaerobic digesters. And the introduction of these specific bacteria resulted the high activity in acetogenic digesters. But at the end, the methane and biogas production trends stabilize after a while. And it can be concluded that we can use repeated augmentation strategy, repeated inoculation strategy to have higher biogas yields in the further studies with this perspective. And thank you for your attention.
Thank you very much for your presentation. Dear participants, do you have any question? I think there is no question.
Okay, thank you so much. It was so clear then. Thank you so much.
Yeah.
Thanks.
Our third presenter is Illyrian Malalori, I think, if it is correct pronunciation. I don't have his CV, but I know his presentation. His presentation title is Dairy Wastewater and Possibility of Ethanol Producing from Cheese Whey Using Process Simulation. Mr. Malalori, microphone is yours, please. But I want you introduce yourself first, briefly, please.
Yeah. Okay. Thank you. Thank you so much.
Thank you.
And as a matter of fact, it has been a long time going together in the Eurasia from 2011, I think, or a little bit earlier when it started. And I am professor at the University of Tirana, member of Academy of Science of Albania, and head of the chemical process engineering group in University of Tirana, Albania. As a matter of fact, there is a group here, are just 4, but we are running a lot of projects regarding to the waste treatment in order to not only to avoid the contamination, environmental contamination, but also we have to see the possibility of reaching the production of some usable products in order to revive, to renew the capacity of their own itself. So let's go into detail. I think my presentation is at the beginning, it is enough. Yes, I'm chemical engineer, and I'm leading the group of process engineering and modeling and simulation of the chemical processes, as well as food process engineering around the region. Starting from Greece to Italy to Northern Macedonia to Kosovo and so on. I'm invited lectures to that country, but for the followers of this conference, it is enough. Mr. Virgili Sinani is my friend for a long time, and we have been collaborating for many years together. So special things that need to be said, that the complex of the participants, of the co-authors. It is a special invited PhD student from University of Ioannina, Greece, Miss Ionilda Lupa. She has collaborating for making possible, let's say, analysis and simulation process during the study. So it is an interesting title, an interesting study, along to other studies trying to use, to revive, let's say, the capacity of energetic value of this waste that coming from different industries. This is from the dairy industry. It is the wastewaters that can be produced not only to avoid the contamination, but also to the ethanol production from cheese whey. So it is really an interesting. Let me see why it is not. It is not. I'm sorry. I don't know. Wait a moment. I hope. Oh, excellent. So. Time and again, I used to say, apologize to the people who are following me in the conference, because in conferences, because I am the worst timekeeper. But anyway, I'm trying to keep my time this presentation. So it is to be said that there are a lot of arguments why we are dedicated to this waste, for the dairy wastewater, and why it is important for the not only for getting rid of contamination, but also to make possible producing some usable and profitable chemicals like not only lactose, lactic acid, but also the derivative of it, that it is ethanol. Of course, it is to be done the first of all the characteristics of these effluents in order to find out which is the congruence of the other contaminants, of the other compounds that is present to the whey, where it is coming out from the process of milk processing. And indeed, there are 3 main points of the technology which has to be this waste need to be collected and then to be storage ready to be treated in the experimental work and in the simulation work. Who is, of course, who is familiar with the modeling and simulation process, they know what does it mean. You have to take a lot of data characterizing this waste first, and then coming into view what it is the way of treating for the designing of a plant usable for application for ethanol production. So there are of course some experimental and process modeling and simulations, I already said before, and then we have made already so far the calculations through the simulation process using a special computer simulation program, which it is a SuperPro Designer. And then we have made some biological treatments through anaerobic digestion. I was happy, and I congratulate for the previous speaker for the a lot of information given to the different areas of the same issues, let's say. Then there are, of course, some conclusions and recommendations. Application of the process simulation for ethanol producing using whey residues from dairy plants seems to be, first of all, important. Two issues that there are not only, this is not only the possibility of using these residues coming from discharging of the dairy products of the milk processing plants. There are some direct uses that, of course, you have to see that there are some byproducts which are already sold in the market, but also there are some indirect uses of this cheese whey, which is a main product is lactic acid, production of bacteriocin, biosurfactants, and ethanol. And we are focusing in that. And I'm going to speak a little bit in details with this subject. If I'm correct with the program already said at the beginning of my presentation, I'm going to first of all, for most of the people who are maybe in the conference hall, know which is the way of treating the milk and what are the catacombs of reaching different products that are going to the market. But we are focusing indeed in 3 points, you see, where it is in the way, and it is coming from the heat coagulation, and when the whey separation through micro filtration of or nanofiltration, we got a liquid that it has to be really necessary to be treated. So from casein precipitation, we have got also a liquid. Then in the second place, it's here, and the third place, from 3 sources, we got a liquid, which is with different type of composition of this. And there is one way that people used to be focused in the industry worldwide. First of all, to profit a protein, which is the most important for the human being health, and they go to just to deliver to the market as a liquid, as in the powder. But also we are going to see that what are some data taken from the experience of international experience. Of course, during removal of milk casein during cheese making, we got a lot of compounds that are really interesting for our own study. So just to show you that it is generated 9 kilograms of whey, cheese whey, for every kilograms of cheese used to be treated. So this is very important in order to let the others know that this is the nutrition value. There is a whey-based beverages on the market. It is colorful packages on the market, delivering other compounds coming from this residue. But we are focusing in indirect uses of whey direction for the production of ethanol, bioethanol. Exactly. I have already mentioned all this usage, but we will focus on that. The residue of cheese and casein production, there are a lot of compounds, a lot of minerals, and in the ashes, you know, it is some inorganic compounds, and we have to be characterized by different protein concentration, 35%, 60%, and 80%. By different studies has been shown that there are a lot of fat, a lot of lactose, and protein. Protein, you may be aware that in the market it is really abundant, and a lot of people used to collect it, to use it for everyday life. So processing alternative seems to be a diagram, process diagram, showing a different application and different biotechnological processing of the cheese whey, but there are also some physicochemical processing that need to go to other directions, and producing powder through lyophilization, and then the whey protein and permeate, which directly goes to producing of ethanol. I'm going a little bit faster because we have to see the results of our simulation. During the processing of food, of course, the whey, the cheese whey, the milk whey, as calling the others, seems to be a lot of things, a lot of processes going from concentration by evaporation or reverse osmosis, and then drying, coming into powder and producing a product that goes directly to the market. But also there are some other kind of concentration and filtration, nanofiltration, which goes to 2 different products, which has different story after the final treatment going to a pharmaceutical product, to renewing to the food industry, like a syrup, and then to animal feeds. I'm sure that if we are having more time in order to explain details, we see that there are a lot of things, a lot of data that need to be shared with the other colleagues which are present online in the conference. But our road goes from this way to the separation, and then for the part of the separation which comes out through metabolites. One, it is a biomass which goes directly to the cell protein in order to profit, also to make a biodigestion, but accompanied by lactic acid, B12 vitamins, and penicillium goes directly alcohol, which is the most percentage of the process, which goes running into ethanol or ethyl alcohol, what it is. Indeed, there is a need for some Saccharomyces cerevisia's yeast in order to profit. So the main role, it is going from the west. Then we make some concentration. Then we made hydrolysis. Then we got the product ready to be fermented.
Mr. Illyrian, your microphone is moved.
I'm so sorry, but I think it's okay now.
Yeah, now it's okay.
Yeah, thank you so much.
But you have 3 minutes.
Oh,
yes.
So sorry. Then let's go a little bit faster. I told at the beginning because so I am a little bit not so good in keeping time, but I'm going a little bit through some chemistry. We go into the Clavieromyces marcianus. It is another strain of yeast we use better than Saccharomyces cerevisia, and then we produce ethanol. Producing ethanol, it is biofuel, and we are going to see that the possibility of simulation this process coming from whey to ultrafiltration, coming directly to the ethanol after dehydration, and we have already now in our lab reproducing this process entirely and producing ethanol and characterizing the ethanol coming into view. So we have made all the material balances. We have made the characterizing of the liquids coming from the milk, the bigger milk industry in Albania. And we have already used the different experimental setups and then characterizing the cheese whey, casein whey, and then start doing the process by experiment. First, starting the experiment through biological treatment in order to make hydrolysis, then simulation. The simulation means mass balance, of course, through the computer, then energy balances, and then we have I don't know why it is time again unmuting, but anyway, first of all, we have to follow all the standard procedure of the Then of course we have done through the exercises for the basic computer program like SuperPro Designer, and we have developed mass balance complete, energy balances, then preliminary design. At the end of the things, after sizing the equipment, the calculation, we have done a lot of reports that we have calculating everything in details according to the procedure of process design. And which is the most important, we have make the process entirely diagram of the process, which has to be followed. You see, at the beginning, it is milk processing, then the cheese, integrated cheese making, then it is butter making. But this is the ethanol, exactly, this is the ethanol sector of producing from cheese whey. then separating from that complex scheme, we in SuperPro design means that we have a reactor for fermentation. Then after reverse osmosis, we got the liquids coming with a value that needs to be fermented, changing the things to the sugar content. Then after the fermentation, we make a centrifugation, then a distillation column in order to increase the concentration of the ethanol produced, and then we can store this ethanol. There are some data now coming from the simulation, and you see the results. There is 17 liters of ethanol for 100 liters of whey, and then some investment that need to be done to be sure that we have secure all the from different let's say potential supplier of the equipment for making possible to produce the ethanol with the plant in reality, in the industrial capacity. And then we have follow all the calculation coming from emission report, from administration. And then it is a kind of a preview, which is the future of our study in order to bring into reality this project for producing bioethanol from cheese whey. So as a conclusion, of course, the benefits, it is understandable, which it is, because it is this chemical precipitation, it is due to a lot of rich in organic materials, nitrogen and phosphorus. And we have been focused on dairy plant operates around 8,000 hours per year, which is a chemical industry normally. And we have produced, you see, 0.002 metric tons of per hour, 95% of pure ethanol. First of all, we produce 60%, and then in concentration to the distillation, to the rectification, to up to that. So producing biofuel, it is a big direction for the positive environmental impact. And for this reason, bioethanol production offers an ideal alternative to whey valorization. Ethanol production is restricted to microorganisms. Of course, we use to change from Saccharomyces cerevisiae yeast to another yeast, more productive and more prospective. In Albania seems to be one of the interesting projects that needs to make, let's say, reality for the economy, being helped by the chemical engineers that has been produced, let's say, all these data. Thank you so much.
Thank you very much for your presentation. Dear participants, do you have any question? I'm waiting for your response via chat if you have a question. Okay, I think there's no question.
Thank you so much. I'm very happy that the project has been already delivered shortly. Thank you so much.
Thank you. Our fourth presenter is Özlem Kaplan. Özlem Kaplan received her Bachelor of Science degree and Master of Science degree in Environmental Engineering in 2015 and 2017, respectively, from Marmara University in Istanbul, Turkey. She is a PhD candidate at Marmara University Environmental Engineering Department, and her presentation title is Heat-Assisted Vacuum Disintegration of Waste-Activated Sludge for Improved Biogas Production. Ms. Ozbayram, are you here?
Thank you for introducing me.
One is yours, please. Okay, go ahead.
Hello, dear participants. I would like to repeat my title again. It is heat-assisted vacuum disintegration of waste-activated sludge for improved biogas production. This presentation is about some part of my PhD thesis. We developed the HAVT process with my thesis supervisors, with Deniz Akyol and Barış Çağla, and we have a patent application for this new technology. Firstly, I will begin with the aim of our study. Then we will look at some underlying information about biogas, anaerobic digestion, and pretreatment methods. After the introduction, I will introduce you to our developed system. And finally, we will evaluate the results and see conclusion. The first aim of the study was to make the disintegration experiments in order to break up activated sludge blocks and explore the microorganisms. Here you can see the structure of activated sludge blocks. It consists of bacterial colonies, filamentous bacteria, EPS matrix, and particulate organics. Activated sludge blocks are broken up to its elements by disintegration methods. The second aim of the study was to evaluate the benefits of the vacuum treatment in terms of heat and electricity production based on biogas enhancement over 5 different scenarios. It was very important for us because, as we all know, that cost estimation is a big part of any technology in engineering. Now let's look at the importance of biogas. Escalating energy consumption increases the demand for renewable energy source. Therefore, we need alternative energy sources. In this sense, biogas is an important renewable energy source with a high calorific value. It has no greenhouse gas emission and it enables less usage of fossil fuels. Biogas is mainly composed of methane and carbon dioxide, and its calorific value increases with the increase of methane percent. So improved biogas expression at the title means that high methane percent in biogas. In biogas production, an anaerobic digester is used, and the digester can be fed with several waste materials having high organic content, such as manure, food waste, and wastewater treatment sludge. In our study, we have dealt with the waste-activated sludge, which is produced at a wastewater treatment plant. However, waste-activated sludge, especially obtained from extended aeration systems, has a very stable structure, which is hard to decompose biologically. Because of this reason, in extended aeration, a pretreatment may be applied to waste-activated sludge before the anaerobic digestion. Now let's see how anaerobic digestion process occurs. The steps of anaerobic digestion are hydrolysis, acidogenesis, acetogenesis, and methanogenesis. Our interest was hydrolysis step in this study. If we look at the hydrolysis step closely, we can say that it is the breakdown of complex organics into soluble organic substances, which are smaller and can be easily decomposed by microorganisms. In anaerobic digestion of fats having high sludge age, the rate-limiting stage is hydrolysis. In order to increase hydrolysis rate and enhance the anaerobic digestion, a pretreatment method can be used. The sludge pretreatment technologies are used to disintegrate the flux and microorganisms and thus increase the solubility, dewaterability, and biodegradability of sludge by changing its physical, chemical, and/or biological properties. They can be mechanical, chemical, biological, or thermal. Our developed system HAVT process may be called as a combination of mechanical and thermal pretreatment methods. Now let's see how we perform the experiments. We have used thickened glass and digestate, which have total solid amount of 4%. The experiments have done in an airtight bottle, which was connected to a vacuum pump. The bottle was mixed with a magnetic stirrer during the experiments to maintain a homogeneous temperature and TS content. With the help of the vacuum pump, pressure in the bottle was kept under atmospheric pressure. At 59 degree, the sludge started to boil at a pressure of 18 kilopascal. The HAVT test was applied for 3 hours. Every 30 minutes, the vacuum pump was turned off and sludge samples were taken for soluble chemical oxygen demand analysis. You can see the characteristics of thickened was and digestate in this table. In this video, we can see a part of the experiments. During vacuum application, the pressure is kept under atmospheric pressure and molecules start to move. Then sludge boils when the molecules have enough thermal energy to overcome the intermolecular attractive forces that hold them together, thereby forming vapor bubbles within the sludge. This enables the disintegration of flux and cell rupture. I can explain the difference of our study from the existing studies in the literature like this way. We have used the synergistic effects of vacuum and moderate temperature thermal treatment to obtain a better pseudo solubilization with a lower energy requirement. In these graphs, we can see the results of experiments. Soluble pseudo increases with the HAVT process for both thickened waste activated sludge and digestate. The increase in soluble COD value is 327% for was and 53% for digestate. The reason of the significant increase in soluble COD values is the breakage of flux structures in the sludge, disruption of microbial cells, release of cell content, and conversion of particulate organics to soluble compounds. Therefore, the anaerobic digestion process will be stimulated, so both biodegradability of the sludge and methane yield will be higher compared to the untreated sludge samples. Moreover, the required time for hydrolysis will be less and the construction cost of anaerobic digestives will decrease. In the second part of the study, an energy analysis was performed for different digestion scenarios in order to evaluate the performance of HAVT on the digestion process, heat and electricity production. These scenarios were built based on total solid loads for a full-scale application, and only digestate characteristics were used in estimations. Sludge flow scheme of the designed anaerobic digestion system coupled with HAVT units can be seen in this figure. In this system, the digestate withdrawn the digester is conveyed to the vacuum vessel after being heated up to 59 degrees with a heat exchanger. After vacuuming at 18 kilopascal and 59 degrees for 3 hours, the digestate is recycled back to the digester. To calculate the energy required for HAVT process, a mass energy balance approach was used. The assumptions of HRT, BS content of raw sludge, methane percent of biogas, and increase in biogas amount after the HAVT are listed here. The energy requirements and biogas-based electricity and heat generation potential of the developed system were calculated in order to evaluate the energy balance of HAVT process. Theoretical biogas and methane production values were calculated based on assumed organic content of the sludge. A combined heat and power system was integrated into the scenarios. This cogeneration provides electrical energy and useful heat by combusting the biogas. Both the heat and electricity production efficiencies of the cogeneration system were chosen as 40% each, and the rest of the energy was assumed to be lost. The generated heat is used for heating the vacuum vessel and the anaerobic digester by heat exchanger, and the generated electricity is used for the vacuum pump. The produced heat and electrical power are directly associated with the production of methane gas in anaerobic digestion and increases with soluble CO2 increment in feed sludge. In this graph, we can see the comparison of different scenarios. Red parts are the heat requirement for HAVT application. Blue parts are the heat requirement for digester heating, and the green parts are heat generated via cogeneration. The generated heat, the green part, was calculated by considering 20% increase in biogas as a result of HAVT pretreatment. Digester heating requirement drops with increasing TS load because the volume to surface area ratio decreases as digester becomes larger. This figure also shows that the heat remaining after the HAVT increases with rising TS load. This graph is also a comparison of scenarios. Here it is shown that the electrical power generated via cogeneration, green parts, extra electricity production, the blue parts, and the power used for vacuuming, red parts. They are very small. Here, the electrical power consumed by the vacuum pump was estimated to be very low with respect to the electrical power generated. Electrical energy obtained from the extra methane produced after the HAVT is highly enough to compensate the energy required for vacuuming. Ratio of electricity consumption to extra electricity production never exceeds the 10%, as you can see from these values. Finally, it was indicated that the heat required to keep the temperature at 59 degrees during the HAVT is less than the amount needed for digestive heating. This is a very important point. The electrical energy required for vacuuming is significantly lower than the extra electricity generated.
Özlem, you have 3 minutes. Okay.
Okay, conclusion part. As a conclusion, I can say that HAVT was very effective in enhancing the digestibility of past and digestate. HAVT can be used as a promising alternative in sludge pire and or site treatment. The anaerobic digesters can be operated more efficiently and may produce more energy per unit volume of sludge to be digested. The 68% of the heat generated via cogeneration is adequate for keeping the temperature at 59 degrees for 3 hours during HAVT. Improvement in biogas amount and methane content is very important in constructing energy self-sufficient or energy positive sewage treatment processes. Thank you for listening. And here I would like to also the organizing committee and my thesis supervisors. I can take questions if you have any.
Thank you very much for your presentation. And I would like to ask the participants for the questions they have. Do you see the questions, Ms. Özlem, from the chat part? There's a question. It's a little bit long. You can read.
Yeah, I can see now. In figures. About soluble chemical oxygen simultaneously. It has shown an increasing trend to understand contents every time, increases every time. No, Emre Oz asked like, have you tried longer times for COD experiments? No, no, I haven't tried the longer times yet. Actually, I haven't tried the longer times yet, but I can consider longer times for next experiments. But thank you for your question.
Maybe you can try further studies, longer time maybe.
Yes, of course. I will take into consideration this contribution.
Okay. Do you have any other question? Okay, I think there is no question. Okay,
Okay,
thank you very much again.
Thank you for this chance to organizing committee, and I wish everyone a good day.
Okay, thank you. Our fifth presenter is Mehmet Akça. Mehmet Akça received his Bachelor of Science degree in Environmental Engineering in 2011 from Bahçeşehir University in Istanbul, Turkey, and his Master of Science degree in Environmental Engineering in 2015 from Istanbul Technical University in Turkey. He is still PhD student and research assistant at Istanbul Technical University in Environmental Engineering Department. His presentation title is Istanbul Technical University Sustainable Bioenergy Production Plant, Preliminary Results from Container Type Digester Utilizing Food Waste Produced at ITU Campus. Mr. Akça, are you here?
Hello.
Okay, the microphone is yours, please.
Okay, I can share screen, I think.
Okay, yeah, you can.
Hello everyone, this is Mehmet Akça from Istanbul Technical University. I'm gonna make a presentation about the small-scale compact biogas plant located at Istanbul Technical University, utilizing the food waste produced at the campus. Our project was funded by Istanbul Development Agency, as you see the number of the project, as well as from the Directorate, and executed by Dr. Mahmut Altınbaş. We named it biogas, inspired from the bee, which is a symbol of Istanbul Technical University. Our plant actually consists of 2 containers and 1 green office, which I'm going to tell you in detail. One of the containers is the digester, or you may call it fermenter as well. And in the other container, the mechanical equipment like CHP units and hygienization tank is located, and in the green office, actually it's the office building of the plant, but we apply a ecosan approach, ecological sanitation approach in the office of the plant. We separate the wastewater streams as yellow water, brown water, and gray water. We also collect rainwater. The gray water and rainwater is mostly used to regulate the solids content in the digester. And we grow microalgae on the photobioreactors mounted on the facade of the office building with yellow water, human urine, with certain dilution rates, of course, which are eventually fed to digester. And brown water is also fed to the digester. Our process flow is actually first the food waste produced at the campus, mainly in the dining hall, yemekhane, and the campus is carried with carts, as you see in the number 1 here, and it's transported via screen conveyor to the grinder for volume reduction. And after grinder, it's transported to the macerator to regulate, to give the desired solids content. And after the macerator, the slurry produced at the macerator is transported to hygienization tank, which is dictated by the regulations in Turkey. It stays there in 1 hour for in 70 degrees Celsius to remove pathogens. And after that, the slurry or the waste, food waste is transported to the digester. Sorry. Dining Hall of Istanbul Technical University has a capacity of 12,000 people. We have actually fed with the leftovers. We fed the fermenter with the leftovers up to this point, like celery, leaf and grass, potatoes, carrots, and other vegetables. And we had to stop feeding due to pandemic because the dining hall stopped to operate in the pandemic period, so we had to stop operating the plant. Small-scale biogas plants are actually common in many parts of the world, and including India, Southeast Asia, Europe, as well as in America. In India and Southeast Asia, the biogas produced is usually used as cooking gas in the kitchens. But in Europe, electricity and heat is also produced. And in Northern America, even the farms which have less than 100 cattle may have small-scale compact biogas plants to produce energy, electricity and heat. Of course, several problems with operating the small-scale compact biogas plants are reported in the literature. One of the most common is the clogging of the inlet pipe, which we don't have. We just transport via spring conveyor after the collection carts. Especially in the Southeast Asian countries, the constructional instabilities in the rainy season is regularly reported. And as well as the dynamics of the problems arising from the dynamics of the reactors are also reported, like solid accumulation in the surface of the slurry. As well as leakage problems are reported. And in some parts, the strict legislative measures may pose a barrier against small-scale, even family-scale biogas plants. Our plant is seen on the figure. The right-hand side in this container, the mechanical equipment is located. On the left-hand side is the fermenter. It's simple to build and mobile. It consists of containers. It's an on-site treatment systems around 50 cubic meter volume. I'm going to present the biogas production results for 1 month because after that the pandemic started and we couldn't feed the fermenter. We tried to keep the solids content in the reactor below 8%. Below 8%, it can be considered as wet type digester in this point. We operated in mesophilic conditions near 38 degrees Celsius, and on average for 1 month we fed 132 kilograms of food waste per day. This is not dry weight, by the way. This is the wet weight. We have CHP unit, as you can see in here. It has 35 kilowatt installed capacity, and we have achieved the I'm sorry. We achieved the 15 kilowatt hours net energy production in the 1 month of operation of the plant. In these graphs, you can see the biogas production, the amount of biogas production per day, and the daily food waste. We couldn't feed the digester every day in 1 month due to several inconveniences within the university. Sometimes the collection cars were not available. Sometimes the wastes were already disposed. And as you see, well, the biogas production seems consistent with the feeding, and the digester is able to produce biogas. We have achieved 672.5 liter biogas production per kilogram VS fed to the reactor. Continuous biogas production can be achieved with continuous loading. We're actually hoping to start continuously operating the plant, but right now it's not possible. I mean, the dining hall is operating, but you know, they're serving the food with the bags, and people eat it and they, you know, dispose it to wherever they want. We cannot collect food waste at the moment. As I said, 15 kilowatt hours electricity energy production we could achieve. And there are electricity cars in the campus. The produced electricity will be used by these cars. There are actually charging stations in the campus, and of course we're not gonna give the electricity to a network here. We're gonna give it to the charging station for electricity cars. And as conclusion, the biogas yields in our container type digester is consistent with our lab scale experiments. This is of particular importance for us because it implies the performance of the 50 cubic meter reactor, because we're doing lab scale experiments like several hundred milliliters, and the results are consistent. System planted at Istanbul Technical University typically occupies less area than other small-scale systems. Most of the small-scale systems, especially in Europe, they're assisted with post-digesters, round concrete circular ponds, and maybe additional hydrolysis units. And we didn't encounter any leakage or construction-related problems in the 1 month of operation. Also, with the hygienization, we could meet the strict legislative measures, and we believe it offers a compact solution, like communities, campus-like communities, or even like small settlements. Right now, as I said, due to the pandemic situation, we had to stop operating the plant, but we're hoping to restart to operate the plant as soon as possible. Thank you for listening.
Okay, thank you very much for your presentation. I hope you will start again in a short time.
Hopefully, thank you.
Do you have any question, dear participants? Okay, there is no question, I think. Okay. Okay, our last presenter is Priscilla Maradini. Priscilla Maradini received her Bachelor of Science degree in Environmental Engineering in 2018 from the Federal University of Icosa in Brazil. She is a master's student in Environmental Quality, Waste Treatment and Resource Recovery at the Federal University of Icosa, Agricultural Department. Her presentation title is Insights to Improve Covered Lagoon Biodigesters in Pig Farms. Ms. Maradini, are you here?
Hello,
Okay.
hello everyone.
Microphone is yours, please.
Okay.
Can you stop screen share, Mehmet? Okay, thank you.
Can you guys see the presentation?
Yeah.
Okay. So hello everyone. Thank you all for being here. Thanks for the introduction. And I'm going to present this research that I'm part of that is entitled Insights to Improve Covered Lagoon Biodigesters in Pig Farms. So in Brazil, the pig farming activity is one of the most important activity in the Brazilian agribusiness. However, this activity generates effluent with a high pollution potential. So the treatment is essential. And one of the alternatives to the treatment of these effluents is the anaerobic reactors. And in Brazil, the covered lagoon biodigesters stand out as the most important anaerobic reactors that we use to treat the effluent generated by pig farms activities. So the covered lagoon biodigesters have some limitations as the operating temperature, the influence of hydraulic regime, and the lack of effluent recirculation. However, there are some advantages as well, as the low costs, the low energy demand, the low sludge generation, the easy operation, and the biogas production. And the biogas can be converted into thermal and electric energy. The biogas recovery has other important uses as well, like they can greater the sustainability of pig farms, can increase the Brazilian renewable energy metrics, can reduce the CO2 emission, and also helps to get to the goals of the Paris Agreement. The pig farming in Brazil, there are some studies that say that the pig farming in Brazil has the potential to generate an amount of 1,750 terawatt hour per year of electricity come from the biogas recovery. However, this potential is very unused here in Brazil. As an example of the state of Minas Gerais, that we have only 34.8% of pig farms use the covered lagoon biodigester to treat their effluents. And from this amount, only few generates energy from biogas recovery. And the low use of biogas energy potential can be due for the lack of regulation and incentive policies for the development of accessible and appropriate technologies in the Brazilian context. So we believe that monitoring and optimization of the anaerobic process as regarding the constructive and operational improvements of the process can increase the organic load removal, the biogas production, and reduces the greenhouse emissions. In this way, this work aimed to present scenarios and alternatives associated with constructive and operational improvements of covered lagoon biodigesters, as well as integrating their byproducts recovery in order to improve pig farm sustainability. So the interventions that we talk here in this research, they came from scientific papers, technical equipment manuals, technical research, and consultation with professionals. And we separate the optimization strategies in two parts: the technology alternatives, including the preliminary treatment, operation temperature, and effluent recirculation, and the byproducts recovery, considering the operation temperature and the biogas energy recovery. So here we are going to show a typical configuration for pig farms using the covered lagoon biodigester. So we have the influent coming from the pig farming activity, and then this influent goes to a sieve or a grid to retain the coarse solids, and the part that didn't retain in the sieve or the grid goes to equalization tank. This equalization tank is used to equalize the flow rate, and then after go to the biodigester. So the treated effluent goes to the post treatment, as can be going to a lagoon, for example, and then to a final destination that can be destination to the soil or a fertirrigation. As a byproduct, we have the generation of the biogas. And the biogas in a typical configuration, it goes to a burner, to a flare, as a mandatory safety equipment. And it's important to say that in this base scenario, the most important heat source to the biodigesters, to the anaerobic process, is coming from the solar radiation. And here we are putting some of our improvement strategies that we considered. So we have the influent coming from the pig farm and going to the sieve. The part that is not retained go to the equalization tank. And then the first improvement is the use of the pre-treatment, that's being the solids removal. After that, we have the swine effluent heating via solar energy. The another one is the recirculation of the treated effluent, and the alternatives for the use of the biogas. So instead of going to the burner, the biogas can pass first to a filter to remove all the impurities, and then the biogas can go to a boiler or to a generator engine to provide electricity, or can be used as a biomethane. And here we have almost the same improvement strategy, but we change the heating the swine effluent for a different form. So the pig farm, we have the influent coming from the pig farm activity and then going to the sieve. The part that's not retained go to the equalization tank, and then to the solids removal as the pre-treatment strategy. And then going to the biodigester, the recirculation of the treated effluent coming from the biodigester. The biogas, the alternatives for the use of the biogas. So we have the biogas going to a boiler or a generator engine. And in this case, we can use this biogas becoming a thermal energy. And then we can use this energy to heat the cold water that will be used to exchange the heat with the influent. And then we have a heated influent going to the biodigester. This exchange heat can occur outside the biodigester or inside the biodigester as well. I'm going to describe a little bit about the improved alternatives that we talked about. So about the preliminary treatment, that's very important to the covered lagoon biodigester operation. Once this reactor operates with a low level of total solids, about around 3%. And also the preliminary treatment decreases the recalcitrant materials, making the microbiological activity better, and then producing more biogas. And we have different methods for the preliminary treatment, but here we put the rotating sieves as an example that has been used a lot in the pig farms that have these improvement alternatives. The operation temperature, so the mainly heating source for the biodigester is the solar radiation on the top layer of the biodigester. So it's very important to choose a dark colors for the dome of the biodigester. Once this specific dome can absorb more solar radiation. And also we can say that even in countries as we have a tropical climate as Brazil, we still see that the covered lagoon biodigester operates at a temperature below the ideal for the anaerobic process. So the effluent heating stands as being a good possibility to increase the heat inside the biodigester and increase the microbiological activity inside the reactor. About the effluent recirculation, so this effluent recirculation increase the degradation of the organic matter once they increase the cell residence time, provides a greater contact between substrate and microorganisms, and it standardizes the effluent temperature. However, this method can also accumulate ammonia and volatile acids and increase the viscosity of the effluent. So it's very important to monitor the treatment, even if we have this alternative or not, but if we have this alternative, it's very important to monitor it. And about the biogas energy recovery, so we can use, if we are going to use the biogas, it's important to filter to get more methane instead of other gases. And then we can use the biogas in boilers, in generator engine, and as a biomethane. And we can use for generate electricity by the generator engine or thermal energy, and then heat the effluent. The economic viability of the biogas recovery depends on local factors, and the investment in machines and the amount of biogas produced favor the large producers instead of the small producers to be worth it to use this alternative. And the impact of biogas recovery depends on the pig effluent composition. So it's very important to do the life cycle analysis of all the anaerobic process and also the biogas production to check the real feasibility to recover the biogas. So as a conclusion, the covered lagoon biodigester is a simple and suitable alternative for pig waste treatment. The optimization of this system requires evaluations of some constructive and operational aspects. The use of strategies to improve the anaerobic digestion process in biodigesters promotes an increase in the treatment efficiency and in the biogas production. The biogas energy recovery is still not very widespread, which highlights the need to disseminate techniques and studies in order to improve the economic viability of biogas energy processes. The creation of regulations and policies may incentive the development of accessible and appropriate technologies for the Brazilian context. And the use of tools such as the life cycle analysis can favor decision-making and comparing proposed alternatives. I'd like to thank for the founding agencies in Brazil, that's CNPq, CAPES, and FAPEMIG, and also thanks to the Agriculture Department of the Federal University of Viçosa. And I would like to thank you all for watching, and I'm here to, if you guys have any questions. Thank you.
Thank you very much. We have any questions, dear participants? I'm waiting for a response, but I think there is no response.
Okay.
I want to thank all of you for participation and for presentation. And we would like to see again at 3 o'clock in the session 2. Okay, see you.
Thanks for your participation.